Thank you everyone for joining us today. First of all, I'd like to make a small introduction about FOS, and then we can start today's webinar officially. So today's topic for this webinar is exploring novel inducers of ferroptosis and related cell death mechanism. It is presented by Professor Scott Dixon from Stanford University, and it is hosted by Professor Rachid Skouta from University of Massachusetts. And then please meet our 5 panelists, Professor Valerian Kagan, Professor Julia Bayer, Professor Jessalyn Ubellacker, Professor Ide Zhang, and Dr. Kenichi Shimada. So I'm gonna, make a small introduction about us. So Ferroptosis and Oxidative Stress, we are the first international journal dedicated to ferroptosis and oxidative stress. And it is an open access peer-reviewed journal published by Science Exploration Press based in Singapore. And then please meet our editor-in-chief, Professor Douglas Green from St. Jude's Children's Research Hospital. So Professor Green's first intention of launching FOS is to advance ferroptosis and oxidative stress research through publishing, scientific dialogue, and global collaboration. And for us, we think one of the strongest strengths of FOS is that we have a highly prestigious editorial board. So we have 37 top scientists from 13 countries, which includes Professor Brent Stockwell, Professor Marcus Conrad, Professor Julia Bayer, Professor Andreas Linkerman, Professor Boyigan, Professor Scott Dixon, Professor Valerian Kagan, Professor Xue Junjiang, Professor Qin Zhong, Professor Barry Hallowell, Professor James Olesman, Professor Navdeep Chandel, Professor Rachid Skouta, and among others. And although FOS is a relatively young journal, but we have actually already published over 40 high-quality articles. And you can see from the screen, it is only a part of our published articles. And you can always go to our website to see the rest of it. So because we think journal is not about only published articles, so we have our communication is really important. So that's why we have collaborated with several conferences this year. For example, we have collaborated with Golden Conference for this year's meeting, cell-based redox regulation and signaling. And also we have collaborated with the Cold Spring Harbor Asia for this year's meeting, Aeron reactive oxygen species and ferroptosis in live dyes and diseases, which will be held in YT Japan this year in November. And also we have collaborated with the second Redox Biology in Health and Diseases conferences. So our goal for FOS is to build a global community for ferroptosis and oxidative research. So if you are interested in FOS or if you just want to consult anything, please contact us through our email fosjournal@scienceexplore.com. If you have any ongoing research projects or research projects, we warmly welcome original research, reviews, or even short communications. Because we are a dedicated journal and we have a very powerful leading editorial board, and we are doing our best to increase our international visibility. And right now we have a relatively rather rapid editorial handling process, and we have no APC until April 2029. So we will be honored if you could consider us as your next contribution platform. And last but not least, please scan the QR code below. You can go to our website and please follow us on our LinkedIn. And if you speak Mandarin and if you have a WeChat account, so please follow us on our WeChat. And that's done with our little introduction, and we can welcome today's host, Professor Rachid Skouta. So Professor Rachid Skouta is a faculty member at the University of Massachusetts. Right now, his research centers on medicine chemistry, chemical biology, and drug discovery with a special focus on ferroptosis and diseases mechanism. So let's welcome today's host, Professor Rachid Skouta.
Thank you for having me. Good morning, good afternoon, or good evening, everyone, depending on when you are joining us from. Welcome to the first webinar of the FOS Global Forum Series. Thank you all for taking the time to joining us today. I can guarantee you it's going to be an exciting discussion and exciting talk. So my name is Rachid Skouta. I'm a research associate professor from the University of Massachusetts, and it's my pleasure to host uh, uh, this webinar. First, let me introduce the speaker today. Professor Scott Dixon from Stanford University is one of the pioneer scientists who discovered ferroptosis. Back in 2012, during his postdoctoral research in Professor Brent Stockwell, together with his colleagues, he identified and named the ferroptosis as novel form of iron-dependent regulated cell death, opening a new direction in the cell death era. Since then, his research has focused on uncovering the molecular mechanisms of ferroptosis, its links to cellular metabolism, and its role in cancer and other diseases. His laboratory at Stanford has made important contributions, I'll cite a few, to identify key regulators of ferroptosis and advancing our understanding, exploring novel inducers of ferroptosis and related cell death mechanism. In addition of our speaker today, we are very honored to have 5 distinguished panelists joining us today. They are Professor Valerian Kagan, the Redox pioneer from University of Pittsburgh, Professor Jessalyn Ubellacker from Harvard Chan School of Public Health, Professor Helia Bayer from Columbia University, Professor Professor Yilei Zhang from Zhangtong University, and finally, very delighted to have Dr. Kenichi Chimada from Harvard Medical School and Brigham and Women's Hospital. So we are very grateful for the 5 panelists for today. We look forward to your discussion and after the presentation by Professor Dixon. With that, it's my great pleasure to turn the floor over to Professor Dixon. Let's welcome the speaker for today, Professor Scott Dixon. Welcome again.
Okay, well, good morning, everybody, or good afternoon, or as Rachid said, good evening, depending on where you are. I'm very happy to be here and speaking at this webinar. I'm grateful for all of you who chose to join the call, especially at times that might not be ideal for everyone. And also thank you to all the panelists, my close colleagues and friends who are here to have a discussion later after we introduce some ferroptosis background and talk about some thoughts here that many of us share. Okay. So my main interest is in cell death and how cells die, and this is an AI-generated image of a dying cell. I think it's quite nice because it captures some of the key features like the permeabilization of the plasma membrane being important no matter how a cell dies. And you can imagine that once you do this, the metabolism of the cell is going to be massively disrupted, and ultimately you're going to get death. And so when I think about what is the definition of cell death, I think about terminal permeabilization of the membrane. And so this is like the common feature of how all cells die. But this is happening to billions of cells in the body every day. We know this homeostatically. Cell death is important. And if cell death is abnormal, this can be a cause for disease. So you can imagine 2 sort of sides to this equation. You can have too much cell death or cell death when it's not wanted, and this could be a cause of a degenerative process of some kind, or you could have too little cell death. And in the case where there's too little cell death, cells that should die are not dying, and then that could give rise to cancer. And so ideally, what we would like to do is to treat cancer, for example. And a question that we've had for many years is how could we best induce cancer cell death? It turns out, as many of you know, that there's many ways to kill cells. Originally, there was one type of cell death, apoptosis, that was worked on for many years by the editor-in-chief of FOS, Doug Green, among many others. And then subsequently, there was the discovery of necroptosis by Jenny Nguyen, pyroptosis, and then also ferroptosis. And that hasn't stopped. There's many more mechanisms that could be added to this list, and we'll discuss that a little bit later in the presentation. But the work here that I'm going to tell you about today relates, of course, to ferroptosis. A key point is that all of these mechanisms, the different names are meaningful to some extent, but what's really important from my point of view is that at the biochemical level, these mechanisms are all distinct. They involve different genes and proteins, they involve different changes to the cell, ultimately all converging on that terminal membrane permeabilization that I was talking about. And because they have these different biochemistries and different mechanisms of regulation, maybe they have also distinct utilities. So maybe some cancer cells might be more dependent or sensitive to one pathway or the other. All right. So ferroptosis, as probably most people on the call understand, ferroptosis is an iron-dependent and oxidative and non-apoptotic form of cell death. And to go back into the history just a little bit, the discovery here was really enabled by a chemical biology approach. And this was pioneered in Brent Stockwell's lab, as Rachid mentioned. So Rachid and I were both in Brent's lab in the late 2000s, early 2010s, and Brent and his lab had previously discovered these two small molecules, Erastin and RSL3, and that they knew that they were lethal to cells. And then when I joined Brent's lab for my postdoc, my project became in part to understand how these molecules were killing the cell. And so to do so, we took a number of different approaches: chemical, genetic, biochemical, cell biological, and so on. But because this is fairly well understood and it's been published for many years now, I'll just summarize some of the key elements that we understand and then move on to some of the current problems in this area. So here is just our cell here on the, let's see, yeah, on the left, here's a membrane. This is a cartoon of our membrane with lipid peroxides being formed in the presence of oxygen and iron. And what can occur normally is that the accumulation of these lipid peroxides is suppressed by glutathione peroxidase 4, a very important enzyme in almost all of our cells. And Marcus Conrad and colleagues have done all the pioneering work to show that GPX4 is absolutely essential to prevent cell death and especially ferroptosis in many cells of the body. But GPX4 on its own can't function. It requires a cofactor, mostly glutathione or reduced glutathione especially. And glutathione itself is a tripeptide synthesized by glutamate, cysteine, and glycine. And those amino acids are acquired by the cell in different ways, but a key way that they acquire them, at least in cancer cells, at least grown in a dish, is through this cysteine glutamate antiporter called System XC. And so what this earlier work really showed was that Erastin is an inhibitor of System XC, blocks the intake of cysteine, and when you do that, it shuts down GPX4, whereas Wan-Seok Yang, a colleague of ours, again in Brent's lab, he was able to show that RSL3 and some related molecules are covalent inhibitors of GPX4. And so they sort of work more downstream in the process to shut down this enzyme. And when that happens, then you get overwhelming lipid peroxidation in the membrane. And this is fundamentally what we call ferroptosis. Now, I just want to highlight that this is a general scheme, but the actual details, which lipids, which peroxidation sites, how does that cause death? This work here, I just want to direct everyone to the many excellent studies of Valerian, Kagan, and Julia Bayer, who really have made it possible to understand what is going on the lipid side here and which lipids in themselves are damaged. And so we follow them in that direction. Okay. So a few years ago, I guess not that long ago, 1 year ago, a year and a half, Jessalyn Ubellacker and myself, we were thinking and pondering, sort of where we are and where we're going with the ferroptosis field, and we wanted to write a little bit about this. And so I think we can summarize some of our understanding in this way, that for many years, we've had these small molecule tools, and these tools have been excellent. Erastin and RSL3 are sort of the canonical ones, but there's been many others discovered since, and those have helped us and many others elucidate very key questions about the mechanism of ferroptosis. I would say that there's still probably much to learn, but that at the cell level, we really have a fairly solid understanding of how you can induce ferroptosis and suppress it as well. However, if you think translationally and you ask, well, how close are we to translating some of this information, for example, to treat cancer? Clearly, we're not there yet. There's been, to my knowledge, to our knowledge, no clinical trials. There's been no drugs that have been advanced into humans with the express purpose of inducing ferroptosis. It's entirely possible there's drugs out there that already do it. We just don't understand that. But as far as designing de novo a ferroptosis inhibitor and putting it into people, that's been a real challenge. And then having the actual clinical trial data that would convince people that this is a viable approach to cancer therapy, I think still remains in the future. And so I'll just talk for a bit about some of our more recent efforts on developing compounds that might be useful for doing more advanced preclinical testing. And then the ultimate goal here, of course, is to get to something that could be used in people. And so this was work that we did together with a biotechnology company here in the area called Ferro Incorporated. They had started with RSL3, and then through a medicinal chemistry approach, they arrived at this molecule, which we call compound 28. And so compound 28 doesn't look like the original RSL3, but I'm just telling you that that was the starting point chemically and sort of thematically. But it retains, excuse me, the same concept of a covalent modifying small molecule that can bind to GPX4 and inhibit its function. The key innovation here around compound 28 was the introduction of chemical groups, such as this adamantyl group, which would stabilize the molecule in vivo. So RSL3 itself has quite poor stability in vivo, as well as solubility, and this compound 28 molecule, through these modifications, it improved on those chemical properties, and that made it possible to use this now for some in vivo studies. And we pursued these. This is work from Jason Rodensel, a graduate student in my lab who is now at NYU. And just to show you a little bit of data, we mostly focus on mechanism, but we do try to do some in vivo work. These are xenograft experiments where we implanted tumors from HT1080 cells into the flanks of nude mice, treated them with or without this compound 28, and then we just looked at tumor size and some other phenotypes over time. I'll just mention also that these studies were done with and without palbociclib, cell cycle inhibitor, for reasons that we don't need to get into today. We were interested in the role of the cell cycle. Okay, so here's one piece of data that I think is really compelling. This is looking at tumors extracted from these mice 3 days after their last dose of compound 28. And what we're looking at here is a one-dimensional SDS page gel where we're blotting for GPX4 protein levels. And in the mice that received oral gavage of compound 28, you can see this shift in the apparent molecular mass, which is consistent with engagement of the GPX4 protein with the covalent inhibitor, and then it alters its apparent migration in gels. And so you can use it as a really nice readout of target engagement, which is something that in a lot of studies is quite lacking. But here you can clearly see that compound 28 is engaging most, or maybe all, essentially all of the GPX4 in these tumors, both on its own and together in combination with a inhibitor of the cell cycle. And so that gives us great confidence that this molecule, when again administered orally, can actually arrive at the tumor site. Excuse me. Then we can also look at a readout of ferroptosis. So ferroptosis, as I said, is characterized by overwhelming accumulation of lipid peroxides. One of the downstream products of this is 4-hydroxynonenal, which is a reactive fragment that's generated as a consequence of damage to lipids. And it's one of many endpoints, no doubt, and it's very technically can be challenging to detect 4-H&E, and there's also no doubt about that. But in these experiments, what we were able to see was that when we treated these mice and then extracted the tumors with vehicle or with compound 28, that the increase in staining for 4-H&E immunoreactivity was quite substantial. And Jason could quantify this across many different fields of view, and overall statistically, but also just visually, we could see that we were increasing the amount of this 4-H&E staining, which, again, is an indication that we might be inducing greater amounts of lipid peroxidation. Okay, so what about the phenotype? Well, when we look at the phenotype, this is the actual tumor sizes over time. In gray, you can see the growth of the tumor. Palbociclib on its own has an ability to arrest the growth of these tumors. Compound 28 on its own also had some effect, although not statistically significant at the endpoint. It was really the combination of both that we needed to see a significant decrease in the tumor size relative to the vehicle control. However, as you can see from these data, we were separating the vehicle control from the compound 28 combination treated tumors, but we weren't really seeing any evidence that, we were shrinking the tumors necessarily. We were just sort of preventing them from growing. And so when we go back to our scheme here of where we sort of stand, do we have a drug candidate here that might be useful, for some more advanced, like I said, preclinical testing? I think maybe. I think it would be nice to imagine using compound 28 in a variety of different tumor settings to understand where we might be able to inhibit GPX4. But also we were getting signals from these experiments that maybe GPX4 isn't the best target. There's a lot of toxicity associated with loss of GPX4, again, as Marcus Conrad and colleagues have shown. And these mice don't really tolerate high doses of the inhibitor. So maybe we need other targets. And I'll just briefly mention, I don't have a slide on it, but of course, the ferroptosis network is much more complicated than the initial scheme I showed you, and new targets like ferroptosis suppressor protein 1, found again by Marcus Conrad and James Olsman's labs, has recently been shown to be perhaps a wonderful target, and that's work by Jess Ubellacker and Thalys Papagiannopoulos. Okay. So where are we going? Well, I guess I would say I'm not sure, but I still have this question. What else could we potentially do here to induce ferroptosis, but maybe in a bit of a different way? And so I want to go back a little bit in time to this review that Brent Stockwell wrote together with many others, where he sort of classified 4 canonical types of ferroptosis-inducing small molecules. So class 1 was system XC inhibitors like Erastin. Class 2 were the covalent GPX4 inhibitors like RSL3. Then they defined class 3 as these inhibitors that appear to target the mevalonate pathway, FIN56 being the canonical example, and class 4 molecules that could engage with iron and in some way, still not totally clear, inactivate GPX4 indirectly or directly oxidize lipids. And FINO2 is the classic example there. And so a few years ago, when people in my lab looked at this, we realized that just looking in the literature, many people have studied Erastin, us included. Many people are looking at RSL3 and related inhibitors, but there's really been very little work done on these other classes of molecules, FIN56 and FINO2. And so we thought there was an opportunity there to learn more, and so we wanted to explore it. And so I'll just tell you some of our work here, which led to the perspective that we published in FOS, and which then led to this webinar. So again, to go back in time a little bit to the class 3 ferroptosis-inducing molecules, this all started with Kenichi Shimada. So Kenichi was a grad student in Brent Stockwell's lab at the time when myself and Rachid and others were there. And Kenichi had done this really clever thing looking for novel inducers of non-apoptotic cell death by screening for small molecules that would kill cells but not activate caspases. And from that, SIL56 was discovered as a very interesting lead molecule. However, it wasn't, in the initial studies, didn't appear to be perfect in the sense that it seemed to be inducing a mixed form of cell death. Okay, so it was causing some degree of ferroptosis, it seemed, but maybe something else. So then, through a medicinal chemistry effort led by Rachid, who is here on the call, Rachid was able to derive this molecule called FIN56, and it was so named because it seemed to have a much purer ability to induce ferroptosis. So the FIN is just for ferroptosis inducer, and the 56 was just kept from SIL56. And you can see, just looking at the structures, that it's largely similar, has the oxime, for example, and then it just has this major difference here, this introduction of the secondary amine, which isn't found in the same position, in the same way in SIL56. All right. And FIN56 seemed to induce a much more, as I said, pure form of ferroptosis. And so we were interested in this, and we were interested in the differences between these molecules. Are they really different? Are they similar? The different studies that had found RSL3, FIN56, Erastin, they're all sort of done in different models and contexts. And so a grad student that just finished in my lab last year, Logan Leak, he did a really careful study side by side. in the same context of all these different inducers. Let's just see how they all look. And so RSL3 is shown here. This is a cell death graph where on the y-axis, this is a metric of cell death where we're directly measuring cell death. 0 means everything's alive. 1 means everything in the population is dead. And then we're treating with RSL3. It's a dose response as well as ferrostatin 1, a small molecule inhibitor that's specific to ferroptosis, QBDOPH, apoptosis inhibitor, and TOFA, an inhibitor of lipid metabolism that whose importance will become clear in a second. So here with RSL3, you can see this is the classic phenotype. You completely inhibit death with ferrostatin 1, but caspase inhibitor has no impact, for example. Same thing with a Erastin analog called Erastin 2. These are ferroptosis phenotypes. Okay, so that's just pure ferroptosis. Bortezomib is a control for apoptosis, and we see a very different phenotype here. Here, ferrostatin 1 really doesn't have much of an effect. It's in the noise, but the pan-caspase inhibitor can block cell death. And so this is apoptosis. SIL56 was different. SIL56 in our hands is not really suppressed by FER1, maybe a tiny bit. And it's this inhibitor of lipid metabolism, TOFA, that has a much stronger effect. And so this phenotype we just will call something else. FIN56 also has sort of a combination in our hands, in our experience, a mixture of different phenotypes where certainly a large portion of the lethality is suppressed by ferrostatin 1, but then also at higher doses, it's doing something else. And so we wanted to understand, what's going on here? What are these mechanisms? And so our updated sort of understanding at the time was, well, ferroptosis is definitely part of it for SIL56 and FIN56, but there seems to be a sliding scale of phenotypes here. And one thing Logan had done, he had been able to identify this analog with the help of a medicinal chemist here on campus called tegavin, which looks similar to SIL56 and FIN56. And we thought we would test to see whether it was more ferroptosis-y or more something else. And what made tegavin especially interesting to us is that this molecule is already in human clinical trials. So it's being developed by this company called Iterion here in the United States for the treatment of several different cancers. And it's in people. It's being tested in people as we speak. And so we thought this was exciting because, as I said to you earlier, we really haven't had any designed ferroptosis inducers go into the clinic, go into humans to be tested. And so if tegavin happened to be inducing ferroptosis and was already being tested in people, that would certainly be exciting. And we wanted to figure that out. The strange thing was that at the time, it had been proposed that tegavin was actually inducing apoptosis. This was in the original work from this field. And so that was entirely confusing because, like I said, we had ferroptosis, something else, and apoptosis. So we went in and looked, and in Logan's paper, just to show you a little bit of the results here, with bortezomib, for example, you can see a very clear signal of cleaved caspase 3, a canonical marker of ferroptosis. You don't see this with RSL3. You don't see this with FIN56. Not surprising. You don't see it with SIL56, just like Kenichi had originally found. And you did not see it with tegavin either. So it didn't appear from this experiment that we were getting markers of apoptosis. And then we did a functional experiment where we overexpressed an anti-apoptotic protein, MCL1, in cells. And when we did that, we could make them far more resistant to bortezomib, which was our positive control here. But there was no difference in their sensitivity to SIL56 or tegavin. And so these are just two pieces of data among several that convinced us that tegavin was behaving like SIL56 and not inducing any type of apoptosis. A third piece of data we obtained using a probe for lipid peroxidation. So again, lipid peroxidation is this hallmark of ferroptosis. And these are experiments we have done with the help of Derek Pratt at the University of Ottawa. So he developed this great probe, STY-BOTP, which you can incubate with cells before adding the lethal compounds and then monitor lipid peroxidation. And we really like it for that reason. So you can see here, for example, you treat with RSL3, measure over time, you can see this increase very clearly in lipid peroxidation. You do this for FIN56, you can see something quite similar, although the kinetics are a bit different, definitely an increase. But if you do this experiment with SIL56 or with tegavin, we are seeing really nothing, no increase in lipid peroxidation in these HT1080 cells where we did the experiment. So it doesn't seem like we were getting this canonical marker in a large extent, certainly not with tegavin. Okay, so then in the functional studies here, tegavin was really pure inducer of something else. There was no suppression by ferrostatin 1 whatsoever, but it was suppressed by this molecule TOFA, which inhibits lipid metabolism. Okay, so our updated understanding would be sort of like Tegavant is doing entirely something else, whereas SIL56 and FIN56 are doing parts of something else. And so what is this something else? We've been trying to figure it out for many years, and we continue to try to figure it out. And what we can say so far is that this is some type of palmitoylation-dependent cell death. And I'll show you a little bit of the results for that. With all of these molecules, we've done genetic screens, CRISPR screens, looking for regulators. And from this, we found, for example, ZDHHC5 and Golgi-7 as being key regulators of SIL56-induced death, ZDHHC20 as being a regulator of Tegavant-induced death, and then for FIN56, the work continues. So what are these? Well, these are enzymes that act as protein acyltransferases. So they add to a substrate protein that has a free cysteine thiol a palmitate. Typically, a palmitoyl-CoA is conjugated, or the palmitate portion of it, by these enzymes. These are the enzymes. They have this cool name, ZDHHC, zinc finger proteins, that transfer lipids onto proteins to get protein palmitoylation. Doesn't always have to be palmitate, but mostly it is. So the family contains 23 enzymes in total. There's thousands of substrates. Some of the enzymes function together with a cofactor, one of which is Golgi-A7. And so what we had done a number of years ago was show that in our cells, ZDHHC5 and Golgi-A7 are forming a complex together. And this is one piece of data. This is from a co-IP experiment where we had flag tagged Golgi-7, and then we had pulled out endogenous ZDHHC5. And so if we use GFP negative control, we don't pull down the acyltransferase. Golgi-A7 we do, and then a mutant of Golgi-A7 that is unable to interact with the membrane, this 2CS mutant, it also doesn't interact with this enzyme. And so this was a novel protein complex because it had previously been shown that Golgi-7 can interact with ZDHHC9, and we did see that, but it wasn't clear that it could interact with ZDHHC5. And in our genetics, it was 5 that was more important for the cell death. And so we could, again, do experiments looking at the cell death where we would take our control cells, they'd be killed. Here, this is increasing cell death by SIL56. The knockout would be resistant. We put back in the wild type. They're killed again, just saying they're complemented, and we put back in this mutant, and it doesn't complement it as well. So we feel very good about this complex. So we can mutate it, mutate ZDHHC5 and Golgi-7, and then we could have some questions. How does this death-promoting complex work? And how does it actually cause non-apoptotic cell death? So just briefly on how it works, another previous grad student in my lab, Martha Carlson, who graduated this year, or this last academic year, I should say, she solved a cryo-EM structure for this complex of ZDHHC5 and Golgi-A7. This is 3.8 angstroms resolution, which is pretty good. And then we got assistance from AlphaFold for positioning the side chains. But what you can see here in green is the ZDHHC5. It's a transmembrane protein. These are the transmembrane segments. And Golgi-7 here is nestled on the side near the active site. And so to make it a little bit easier to understand, here's a cartoon of what we think this is looking like. In blue here is ZDHHC5. The DHHC, this is the active site of the enzyme. And then what Martha was able to show is that there's 2 regions, one on ZDHHC5, this RNYR, and one on Golgi-7, this RDYS, that form key interactions between the 2 proteins and probably act to stabilize it. And so that was interesting because these are distal from the active site itself. And so we had this hypothesis that maybe this is involved in stabilizing the complex and necessary for cell death. And so here's a super busy experiment. I'll tell you what it is, and then I'll show you a little bit of the data. This is an experiment where we took knockout cell lines for ZDHHC5 and then reintroduced various mutants that impact amino acids that from the structure appear to be essential for function, and then asked how this affects both interaction with Golgi-7 biochemically on the left-hand side and cell death induction by imaging on the right-hand side. So for example, in these control cells, they're killed by SIL56. These cells that are mutant for Z5, but not transfected with anything, they're resistant to SIL56. This is what we'd found. Then if we complement them with the wild type enzyme, now they're killed again, as you'd expect. On the other hand, if we introduced mutations into this 4 amino acid motif to 3 alanine substitutions, we could no longer interact with Golgi-7 biochemically or complement the cell death. And in fact, what Martha was able to show is we could get all the way down to a single amino acid, this terminal amino acid here in this RNYR motif, mutate that to alanine, and we would lose the interaction with Golgi-7 and the induction of cell death. And when we look in Martha's structure, what we saw is this arginine 147 here, which was that one single mutation, forms what we think is a very key interaction with this tyrosine 76 on Golgi A7. And this cation pi stacking interaction is what's holding together this complex. And when that complex is together, it can catalyze some sort of lethal effect in the cell that's treated with SIL56. Okay, so that's how we think it works. But how are we actually getting the non-apoptotic cell death? Well, we don't know. These enzymes, again, transfer palmitate onto substrate proteins. So presumably, it's the palmitoylation of one or more proteins that is lethal to the cell in response to SIL56 and tegavant. But which proteins are important for this lethal effect? I can honestly say we don't know. However, Albie Joseph, a grad student in the lab currently, he wrote this recent mini review in FOS where he's described sort of our current understanding of how these class 3 ferroptosis inducers might work, and he's proposed some models to explain how they could function. So I encourage everyone to check it out. Albie did a fantastic job putting this together. So some conclusions here to wrap up. We think that these oxime-based molecules like SIL56, FIN56, and tegavant, they can be quite potent inducers of cell death, especially non-apoptotic cell death. In the case of tegavant, it seems like it's safe for using in animals and in humans, which is quite exciting. I mean, there's obviously always a therapeutic window, but it's already being used in people. And so we're quite excited to see what comes out of that. And it's possible that in some cases, maybe it's even a good thing, like with SIL56, that you get the induction of multiple modes of cell death. Perhaps that will turn out to be beneficial if we could advance that kind of idea further by causing ferroptosis and another type of cell death, and maybe together that will be, evade resistance mechanisms and so on. Back to these classes of molecules, I just encourage everyone, I think we all love Erastin and RSL3. I certainly still do. But there's lots of work, I think, that can be done on these other molecules that have come out of related screens. And so I think just in general, I'm excited myself and encourage others to think more about these other classes as well. So can we get to a clinical grade drug? I hope so. I think some of these oxime-based molecules give us a different approach. It's certainly different from RSL3, might act in interesting ways. I know there's other people on the call here that have exciting data related to these classes of molecules, and hopefully that'll be published soon. And with that, I'll just thank everyone who's in my lab currently. I think I've highlighted along the way the people that have done most of the work that I've described, and as well as our former members, and then all of our collaborators over the year. And thank you all. And I guess we'll start the discussion.
Thank you, Professor Dixon. That was very outstanding presentation. We'll now move into the discussion and Q&A session. While we're waiting for some of the questions coming on, I'd like to open the floor to our invited panelists. I'll start with just a thought. As the field moves toward clinical translation, where should we invest our effort? You mentioned during your discussion that developing better GPX4 inhibitors is one, or going toward an FSP1 inhibitors. And another thing about that, currently in clinical trial, tegavant or Vivint is an exciting topic. So I just want to open the floor for our panelists. At no order, you could unmute and share your thought about where should we go for the next 5-10 years from now.
I'm excited to hear from the panelists, so I'd encourage them to speak up.
So Valerian can go ahead first. I saw his hand up.
Yes, thank you so much, Scott. This is phenomenal, as always, kind of brilliant talk and great data, admirable. Well, I'm here more like a redox biology guy. And a redox means that there are two reactions always, oxidation and reduction. And so when you think about targets for killing cancer cells, so one always has to keep in mind that there are conditions where something may be very protective, but there are limited conditions when this protection is over. And this relates to many mechanisms, including the one which has been renamed as FSP1. So because it relies on the availability of coenzyme Q. And then if there is no coenzyme Q, guess what will be happening? What will be happening is that this NADPH-driven electron flow will go to the next most important target, and this target may be oxygen. So now, having said this is just to mention, in exactly the same way, well, ferroptosis is operationally defined as accumulation of phospholipid hydroperoxides. But phospholipid hydroperoxides, if you don't have redox active iron, so they will be stable as a brick in cells and will do nothing. All right. Finally, my point 3. So if you want to cure cancer, so there are at least 2 things that you have to think about. One is that you want to kill as many cancer cells as possible. But 2, if you kill them ferroptotically, so then ferroptosis is nothing but a leaky type of cell death. And now the microenvironment of many cells in tumor will be prone to proferroptotic influences. And one of them is immunosuppression. And some of the cells, like T cells, are extremely sensitive to ferroptosis. Okay, and some of the cells that are immune cells, like myeloid-derived suppressor cells, they're very resistant to ferroptosis. Okay, so now how do you put all these 3 items together? Maybe it was too long, sorry. So, but how do you want to selectively eliminate cancer cells and leave useful cells that could fulfill immune functions healthy and not prone to ferroptosis? That was the end
I
of the
can
question.
echo. Fantastic. Yes, I just want to echo off of that and reiterate that I think that this leads us to the frontier of the microenvironment and the immune functions, as was just mentioned. And I think it's a really exciting opportunity for the field to look into incorporating how those microenvironment factors can influence the perturbations on systemic systems. And so I'm really excited to see work coming out of that space and looking forward to the future of being in the field targeting microenvironment biology as well.
Very well.
I guess,
Please.
Valerian, what do you think of, there's been efforts to make like ADCs or molecules that are more complex than a simple small molecule that can target the tumor cell specifically. Could that be a way to get around this issue of destroying the immune cells at the same time? Maybe. I mean, I don't have any data, but certainly I've seen the papers and they look intriguing.
Well, that's what we try to do, because we know that the microenvironment of solid tumors is highly proferroptotic. And why is this happening? But it may be enough that some of these cells die ferroptotically, and then because this is kind of a leaky type of cell death, all the contents is spilled out, and so then all immune cells are prone to ferroptosis. So now we have a paper now. It's essentially, I'm talking to you about the work mainly done by Dmitry Gavrilovich at AstraZeneca, and we help them a little bit with this. So, but what comes out of this, that we have to understand what are the mechanisms that can make cells of immune system more or less resistant. T cells are highly susceptible to proferroptotic environments, they go dead. And that's what causes immunosuppressive phenomena. So on the other hand, cells like myeloid-derived suppressor cells, they're highly resistant. And the way they're highly resistant, so that they secrete extracellular vesicles enriched with oxidized lipids. And that makes them less highly resistant to ferroptosis and enhances immunosuppression. So my point is that if you only want to kill tumor cells, it's a good, noble goal, but it may be not enough. And just as you said, a combo therapy where you target one group of compounds to cancer cells and another one to innate immune cells, that may be a good approach. And actually, we jointly with Columbia and Pitt, we have an effort to differentially deliver proferroptotic cells into cancer and antiferroptotic or protective agents into immune cells. Yes.
Very well, thank you. Professor Hilya Bayir, can you comment on combinatory therapy? Shall we focus on dye therapy? How about tri-therapy? What's the side effect or toxicity of multiple drugs at the same time? Thank you.
Thank you for the question. Before that, being a clinician, I think one of the important points Scott made is when we're thinking about the clinically applicable drugs, we really need also clinically useful markers of therapeutic effect, right? Target engagement, therapeutic effect, biomarkers of not only ferroptosis, but also the treatment therapeutic efficacy. So I think in that space, we have a lot of work to do and to make the at least lipid peroxidation markers to be much more available, easier to recognize and easier to detect, and in that way. And I think that we have a lot more work to do. In terms of combination treatments, I think what Dr. Kagan talked about, really targeting different populations, cell populations, especially in the solid tumors, right? In that area could be important. And I was intrigued when Scott was presenting the tegavivint that it was in the clinical trials of carcinoma, leukemia, and dermoid tumors. And I was thinking, they're just so different, it seems. What are some of the mechanisms that are common? I think that we just need to have mechanistic targeting. And going back to 1970s, when leukemia was a deadly disease in children, they started adding things. It was first steroid, then vincristine, then another medication. So within 20 years, that almost lethal disease became 80% survival. And then now it's beginning better and better. And I think one other point I want to make is the dietary manipulation. And that goes into the phospholipid, the lipid structural changes, right? Because it's the, we have shown arachidonic acid and adrenic acid containing phospholipids, but also the PUFA, polyunsaturated fatty acid containing lipids in general are much more sensitive to oxidation. And in addition to then combining them with redox sensitizers. So those might be, I think diet is also very important. And some of that is being done, I know, by Brent and others. Those are my thoughts.
I'd like to hear the other panelists for now. 30 seconds each for Kenichi and Zhang, please. I wonder if you can comment on GPX4 degradation. Do you feel, for example, the scaffold we have seen today are a direct degrader? And for chemotherapy, if anybody can also comment on radiotherapy. I know some people are interested here about radiotherapy, some of the a combination radiotherapy.
I can comment briefly on degradation. Well, it is a like part of the mechanism that the Fin56 induces ferroptosis by degrading GPX4, but I'm not sure like that's a viable mechanism in terms of or like more plausible mechanism for the success for the clinical trial. So mechanistically, it would be an interesting direction to explore new scaffold. But I guess that we don't have like too much data to that are accumulated to explore that direction. And I also want to just add this again, what other panelists were talking about in terms of like what is needed in terms of the clinical success. So it is very important to target the cancer cells, obviously, but in terms of the long-lasting response of the drug in human, I guess like immune component is also like very essential. And then like our exploration of the or particularly the optimization of the drug is usually like finely tuned to really target the tumor cells, how the tumor cells become more sensitive. But I think it's also very important to at the very early stage of the drug development to explore how the immune like activation in terms of like increasing the antigenicity is more like optimized, as well as finely tuning the tumor sensitivity landscape so that we can better target or activate both like tumor killing as well as immune activation.
Very nice. Anyone want to comment on radiotherapy? And I have a question for the speaker following this. We can get to Valerian after, if you don't mind. Okay, if there is nothing about radiotherapy, I'm going to go with one of the questions to our speaker.
Can I say, I don't know if we heard from Eli. Did you have any
No,
comments?
I would like to hear from you. Yes, please. Go ahead, Zhang.
Okay. Okay. Thank you. It's really nice to see Scott this time. I bet last time we see is 2 years ago in Suzhou. Oh yeah, and it's really nice to hear your talk again. And I think from your previous publication, we already know that there are different types of ferroptosis inducers. And also, I think based on right now, I noticed that you show data that uracil is the most popular ferroptosis inducers used in the field. And the second one is SF3. I think maybe there are, and also I noticed this phenomenon around our community. And also I think the major reason is because uracil compared with SF3 or BACE4 inhibitor is relatively a little bit moderate, not that strong as potent as SF3. Is that the reason that most researchers choose to use, type 1 inducers, not with type 2 inducers? And also, there are many other drugs, natural drugs or chemical molecules have been found to induce multiple types of cell death, including ferroptosis or apoptosis. And I think is there a key? I'm not sure whether how do they claim it's ferroptosis inducers. Most time I see the data, maybe they just sensitize to ferroptosis, but not directly induce ferroptosis. But there are many publications claims their drugs or molecules can induce ferroptosis. Eventually, I think the publication is more and more complicated. So I think your talk really give us a clear explanation of how these inducers works. And also in terms of radiotherapy, yes, I do believe that ferroptosis is a key cell death mode involved in the radiotherapy, and also our study and including others always focus on this field. And also for immunotherapy, there are many studies too. And I really expect there are more ferroptosis inducers jumped into the field so that we can develop more clinical use drugs to advance the ferroptosis study in clinical. And I'm really enjoying this study right now, and I think ferroptosis is already my career, and I will keep doing that. And I really and also I read every professor's and panelists' papers in the past 8 or 10 years. And really nice to see you guys. Thank you.
Thank you so
Eli,
much.
but radiation induces mostly directly apoptotic death, not ferroptotic death. So how do you put together a radiation-induced, if you want to develop the concept of ferroptotic killing of cancer cells, how do you put a radiation? That will be by definition a combined type of death.
Oh yeah. Ferroptosis mono-induced apoptosis, and also other types of cell death. And from our previous study, we noticed that some cells are more prone to happen to ferroptosis and also apoptosis as well. And some cells that are resistant to ferroptosis are also resistant to radiotherapy. And I believe, I think ferroptosis, of course, is not the major cell death. It's not it's only the solely cell death type involved in radiotherapy. But in some cells, if they are resistant to ferroptosis, they definitely resistant to radiotherapy. This phenomenon is very strong. That's why we still believe in certain contexts, maybe the tumor cells they are resistant to ferroptosis, they can be resistant to radiotherapy. If you combine ferroptosis inducer, you can strongly overcome the radio resistance. And this is one, and also the ferroptosis inhibitors, including lipostatin and ion chelators, we notice that they can strongly prevent cell death induced by irradiation. Of course, I do think the lipid peroxidation is also happened during radiation. And also, I'm not sure is there a cell type specific manner, but at least in some contexts, I do think radiotherapy can induce ferroptosis. And if you combine ferroptosis with radiotherapy, it would largely overcome radio resistance. That's it. Thank you.
Thank you so much. We have a couple minutes to get one question or two from the audience. Thank you so much, audience, for asking multiple questions. We might not reach to your question. I want to apologize for that. This is a question for Scott. How to explore spatial transcriptomics to show ferroptosis? This is from Vanny.
Thanks for the question. I'm not an expert in that. I think Julia made the key point here that we probably need better markers for ferroptosis. If you're going to use spatial transcriptomics, like what genes would anyone consider as being canonical for ferroptosis? I think it would be tough because I don't think there's like a ferroptosis gene, right, that gets induced. So then you're looking at a signature. Could I imagine a signature that's kind of consistent with ferroptosis? Maybe, but I think it would be hard. But again, Julia, you probably have better thoughts here.
Please go ahead.
I think I agree with Scott that it has to be a panel. I don't think that it is, and it's complex. It's 3 huge biochemical areas: thiols, lipids, iron metabolism. It's a very complex system. So I do agree. I think that it has to be a panel.
And if I may add to the point of spatial, beyond transcriptomics, I think the opportunities in spatial lipidomics and metabolomics are really helping us to be able to extend into this space because with the spatial aspect of being able to redefine ferroptosis in terms of where the surrounding vasculature, immune cells, different types of immune cells within that space of lipids spatially could be an area of opportunity that we could further explore too as that technology is being advanced for oxylipidomics with spatial capacities.
That's
Absolutely.
fantastic.
Yes.
Valerian, want to add something?
Well, I want to add to this that so one way to go is to make a list of very specific genes and their products which will or will not be more or less pro-ferroptotic. But another thing is to forget about all this and say, what is ferroptosis? It's essentially a redox active environment in which hydroperoxy lipids decompose and give products. Well, years ago, when we started this work with Hewley in our labs. So we used electron paramagnetic resonance. And this machine, what it measures is radical formation from non-radicals, molecule precursors, okay? And so if you have redox active iron or redox active copper, so you can see formation of these radicals by EPR. And you can use a very sensitive measure of this redox disbalance. Like if you add vitamin C ascorbate, it will give a very characteristic radical signal in EPR spectra. So what is the carrier of this disbalance? That's a big other question. But distinguishing redox disbalance from redox balance is relatively very easy. I'm surprised that the EPRologists did not jump in ferroptosis on this very, very interesting opportunity.
Thank you so much again for this exciting conversation. Unfortunately, we're close to the hour of our meeting. So thank you once again to Professor Dixon for this outstanding presentation, to all of our distinguished panelists for the engaging discussion, and for everyone who joined us with the questions. I apologize for not covering all your questions, but we will reach out to you individually and give you some of the feedback. We hope you found today's webinar informative and inspiring. On behalf of FOS, thank you for being with us, and we look forward to welcoming you to the next webinar of the FOS Global Forum Series. Have a wonderful day or evening or whatever where you are. See you in another webinar.